In this unit, we will study how genetic information is inherited, how genes behave on chromosomes, how DNA works at the molecular level, and how genetic variation is maintained in populations. This is an important unit for CUET PG because it connects classical genetics, molecular biology, and evolution. Our focus will be on understanding concepts clearly so you can solve both theory and numerical questions in the exam. Mendelian genetics core laws. Mendel explains how traits are passed from parents to offspring through genes. The law of segregation says that allele pairs separate during gamete formation. So, each gamete gets only one allele. The law of independent assortment says genes on different chromosomes assort independently. These laws explain common ratios like 3:1 in monohybrid crosses and 9:3:3:1 in dihybrid crosses. Test crosses are especially important because they help identifying unknown genotypes. Gene interactions. CUET high yield topic. Sometimes inheritance does not follow simple dominant recessive patterns because genes interact with each other. In incomplete dominance, the heterozygote shows an intermediate phenotype. In codominance, both alleles are expressed, like in ABO blood groups. Epistasis happens when one gene masks another and changes expected ratio. Pleiotropy means one gene affects many traits, and polygenic inheritance involves many genes controlling one trait, like height. Genetic linkage. Genes located on the same chromosome tend to be inherited together, and this is called linkage. The closer two genes are, the less likely they are to separate during meiosis. Scientists measure gene distance using recombination frequency, where 1% recombination equals 1 centimorgan. Linkage helps us create genetic maps and understand inheritance patterns. Crossing over occurs during meiosis, when homologous chromosomes exchange segments. This creates new combinations of genes called recombinants. Recombination frequency is calculated to measure distance between genes. Sometimes double crossovers occur which can affect measurements. Concepts like interference and coefficient of coincidence help explain these events and are important in genetic mapping problems. Structural mutations change the arrangement of chromosome segments. A deletion removes a segment. Duplication repeats a segment. Inversion reverses a segment. And translocation moves a segment to another chromosome. These changes can affect gene expression and may lead to disorders or abnormal development. Numerical chromosomal changes. Sometimes cells gain or lose entire chromosomes due to non-dysfunction during meiosis. This results in abnormal chromosome numbers called aneuploidy. Examples include Down syndrome, Turner syndrome, and Klinefelter syndrome. Understanding how non-dysfunction happens helps explain these conditions. Cytoplasmic inheritance. Some genes are not in the nucleus but in organelles like mitochondria and chloroplast. These genes are usually inherited from the mother because the egg provides most of the cytoplasm. This type of inheritance does not follow Mendelian ratios, as is common in mitochondrial disorders. DNA replication copies genetic information before cell division. Helicase unwinds DNA. Primase makes RNA primers. DNA polymerase adds nucleotides. DNA polymerase I replaces primers, and Linkage join fragments. Each enzyme has a specific role, and together they ensure accurate DNA copying. DNA replication is semi-conservative, meaning each new DNA molecule has one old strand and one new strand because DNA strands run in opposite directions. Replication occurs continuously on the leading strand and discontinuously on the lagging strand. DNA polymerase also checks for mistakes to maintain accuracy. Transcription is the process of making RNA from DNA. RNA polymerase binds to a promoter, builds the RNA strand, and then stops at termination signals. Prokaryotes use one RNA polymerase, while eukaryotes use multiple types. This process is the first step in gene expression. In eukaryotes, the RNA made during transcription is modified before use. A cap is added to the 5' end, a poly-A tail to the 3' end, and introns are removed during splicing. Alternative splicing allows one gene to produce different proteins. Translation is the process of making proteins using mRNA. Ribosomes read codes and tRNA brings amino acids. Translation starts at AUG and ends at stop codons. The genetic code is universal and redundant, meaning multiple codons can code for the same amino acid. Gene regulation. High yield topic in CUET PG. Cells control gene expression using regulatory systems. The lac operon is activated when lactose is present, while the TRP operon is turned off when tryptophan is abundant. These systems help organisms respond to environmental changes. DNA repair mechanism. DNA can be damaged by radiation or replication errors. So, cells use repair mechanisms. Different systems fix different types of damage, such as mismatched bases, small base changes, bulky lesions, or broken DNA strands. These repairs maintain genome stability. Hardy-Weinberg equilibrium describes how allele frequencies remain constant in an ideal population. The equation is p power q plus 2pq plus q power 2 is equal to 1, predicts genotype frequencies. If real populations deviate from this, evolution is occurring. Forces of evolution. Allele frequencies change due to mutation, natural selection, genetic drift, and gene flow. Small populations are especially affected by drift, while selection favors beneficial traits. These forces drive evolution over time. Final revision. To prepare for CUET PG, focus on understanding concepts clearly and practice numerical problems, memorize important ratios, understand enzyme functions, and learn how processes connect with each other. Practice improves speed and accuracy. Preparation strategy. Effective preparation includes regular revision, solving previous year questions, taking mock tests, and clearing doubts. A structured study plan helps build confidence and improves performance. Consistent study, conceptual clarity, and regular practice are key to success in CUET PG. With strong understanding and disciplined preparation, you can perform well in molecular genetics.